4A, types of experimental design. So first let's talk about the parts of an experiment. Alright, so something called a factor. It's also the explanatory or the independent variable. So you already know what this is, it's just another word for it, okay? The independent variable, the thing that we are changing in our experiment to see if it affects something else, that is called the factor. Alright, the level is when you have the factor can be changed in different ways. So like if it's an experiment to test a new medicine, it could be different doses. So maybe one is like 200 milligrams and the other one is like 500 milligrams. Okay, those would be different levels of the same factor. Alright, so the same medicine, we're not changing the medicine, we're just changing the level. Alright, and then the treatment is the combination of factors and levels that you give someone. So in the medicine example, if we give one group the 200 milligram medicine and then we give the other group the 500 milligram pill, then those are the two treatments. Okay. Dependent variable, we already know that. Response variable, okay, that's the thing that we're seeing if the factor changes it. So if I give someone this different medicine, will it change their symptoms or will it cure their disease? Right, and then experimental units. These are the subjects, the things that are getting the treatment. So it could be people, it could be plants, it could be animals, it could be objects, it could be anything. All right, whatever is actually getting the treatment in your experiment is the experimental unit. Some other vocabulary that we need to know. A control group. All right, a control group is either given no treatment at all or they're given a placebo treatment. So if we take the medicine example, maybe we give a control group nothing and just see how their disease responds. Okay, or maybe we give them a placebo, which would be like a fake pill. It has no medicine in it, but it looks exactly like the one that everybody else is getting. All right, just to see what happens. Because there's something called a placebo effect, which means people react favorably even though they've been given no treatment. So they're taking this pill and there's no medicine in the pill, but they report that they feel better. All right, just because they're taking something, they don't know it's not really medicine, and it makes them feel better. All right, and then we have to look out for in experiments something called a confounding variable. All right, and it's a variable other than the one that we're giving. So in this case, we're trying to test the effect of this medicine on a disease, and if there's anything else in the experiment that could affect their disease, that's a confounding variable because we don't know whether the effect on the disease is from our treatment or from this other variable. All right, so when we design experiments, we have to try as much as possible to make sure that no confounding variables can get into our experiment. All right, that the only thing that's going to change our response variable is the factor, the treatments that we're giving. All right, so 3 elements of a well-designed experiment. When you're designing an experiment, you want these 3 things to happen. First is control, which means eliminating what we talked about, the confounding variable. So your experiment is controlled, and only the one thing that you're changing is the only thing that's going to affect the response variable. All right, randomization, randomly assigning subjects to different treatment groups. Okay, this is important. We have to randomly assign people because like we learned in the first unit about sampling, the way we pick our sample can skew what happens with the results, and the same thing can happen in an experiment. So we want randomization as much as possible. And then replication. Can your experiment be repeated? If someone else did your exact experiment following your exact steps, would they get similar results? All right, that's what we mean by replication. So for this class, we're going to look at 3 different types of experimental design. A completely randomized design, a randomized block design, and then what we call matched pairs design. So in a completely randomized design, right, the subjects are assigned to treatment groups by random selection. So if I have 100 people in my group, I draw random numbers, and the first 50 random numbers that I draw get one treatment, and the next 50 random numbers are in the control group, and that's it. All right, this is the simplest, this is the easiest to do. You just give everybody a random number and pick them. Okay, but what happens sometimes with a completely randomized design is if your sample has some interesting characteristic about it. So like let's say there's more women than men in your sample. All right, just by randomly picking, it could end up that all of the males end up in one group. All right, and we don't want that because we want to be able to compare across the groups. All right, so completely randomized design works best when we have a large sample size. All right, because if we have a larger group of people, there's less chance of all of the males being ended up in one group. All right, and a larger group of people tends to represent a population better than if we just picked 10 people. Okay, the second is a randomized block design, and this is very similar to, if you remember, when we did stratified samples. All right, so we divide the subjects into blocks. A block could be age, a block could be gender, it could be, depending on what our experiment is, our block could be income level, it could be any kind of characteristic that we could divide the subjects by. All right, and then within each block, they're randomly assigned to a treatment group. So if I split them apart by gender, and then I take all the women and I randomly assign them to different groups, and then I go to all the men and I randomly assign them to different groups. All right, so just like a stratified sample. And here's an example of what it looks like. Okay, so we split, we're going to test how students do with no practice test and with a practice test and how they do on the final exam. All right, so we're going to split them up by year. So we're going to take all the juniors and split them up between no practice and practice. And then we're going to take all the seniors and split them up between no practice and practice. And then everybody takes the exam and we compare the results. All right, then there's matched pairs. Now, in matched pairs, we have to go through and specifically pair up two subjects that are very similar. So they're like the same age, the same gender, the same income level, maybe the same height and weight, or very, very close. All right. The classic example that I always see of matched pairs is they take identical twins, all right, and use those as the pairs, and one group gets the treatment and the other group gets the control. So if you take twins, one twin gets the medicine, one twin gets the placebo, and then we see how they react. All right, so we're trying to pair people up that match as many characteristics as possible. That's one form of matched pairs. All right, the second form of matched pairs design is when we have one subject that gets both treatments. Okay, so an example of this might be if we're going to have people run just drinking water and run drinking Gatorade and see if they perform better with the Gatorade. All right, we can't really compare two people's time, like one person running with water and one person running with Gatorade, because one person could be a better runner than the other, and that's why their time would be faster. So in this case, we would do a matched pairs design where they would run twice. One time they would run, the same person would run drinking water, and then the same person would run again drinking Gatorade, and we would compare their times. All right, and when you're doing this form of the matched pairs design, it's important that the order of the treatments be randomized. So everybody can't run first and drink water and then run second and drink Gatorade, because maybe they run slower because they're tired, or maybe they run faster because now they know the course and they're warmed up. So you have to randomize that part of it. So some people will run first with water and some people will run first with Gatorade and then switch. All right, so that is it for 4A, types of experimental design.